Today we will discuss about the growth inhibition and apoptosis. Apoptosis means it's a kind of in apoptosis we will observe the nuclear blubbing, that means nuclear fragmentation will happen. So that is this is 1980 kind of a Nature paper, first Nature paper, where cell will invaginate into some kind of these structures and nucleus will fragment and will go into the different parts of the cell. So apoptosis, DNA fragmentation takes place and the DNA fragmentation factors will does that. This is the 80s research, high mobility group of proteins also. This is my paper in 2010, when compound treated cells will undergo the DNA damage. So the DNA which has to be intact will undergo the damage and the DNA will be the like a ladder like structure will be formed. So how to detect the DNA damage is by Thionel assay, terminal deoxynucleotide transferase mediated deoxyuridine triphosphate Nick labeling. So this is a kind of ready-made kit will be available, and then the protein that regulate the apoptosis are cysteine aspartic protease called caspases. So when they get activated, then only the apoptosis of cells will take place. Suppose you take the cancer cell and add some kind of anti-cancer drug, apoptosis will happen. So initial apoptosis or initiators are caspase 2, 8, 9, 10. These will start the apoptosis and will be executed by caspase 3, 6, 7. So beautiful. So during apoptosis, human apoptosome formation takes place. What is apoptosome means? If you say this, the apoptosome complex when involves the caspase 9, EPAF, and then cytochrome C, this kind of complex is called apoptosome complex. So apoptosis of two types: extrinsic pathway mediated by CD95 TNF receptor and then TNF receptor, trial receptor 1, trial receptor 2. So when the TNF will bind to the TNF receptor, then it will activate the caspase 8 and apoptosis. So it's a kind of external. Similarly, Fas ligand bind to the Fas receptor and activate the pro-caspase 8. And the internal means the stimulus present in the inside only. That's so that means internal caspase is mediated by cytochrome C and mitochondrial dependent. Cancer stem cells coming to the